the proteome. We're going to start off with translational regulation, and then we're talking about post-translational regulation. Make sure, as per usual, you do your reading. So this class, we're actually going to start off with talking about protein unfolding and the proteasome. The proteasome destroys unfolded proteins. Now, both of these are important in a number of different diseases, such as Alzheimer's disease. Some of you may remember this slide from Bio 130. So in Bio 130, we said that Alzheimer's disease is an incurable neurodegenerative disorder. It's generally diagnosed in people over 65 years old. There are about 21 to 35 million sufferers worldwide in 2010, and it's predicted to affect 1 in 85 people globally by 2050. Now the symptoms are going to be quite variable depending on the patient. It's not always going to be the same, but in general, this is what we see. We see people start off with some memory loss. It progresses to confusion, irritability, aggression, and mood swings. Gets worse, these people get language breakdown. Gets worse, you get a progression to long-term memory loss, and eventually death. So what's happening in Alzheimer's and other similar such diseases is that some proteins are not folding properly. The proteasome, which is what we're going to talk about in this particular class, in your cells should remove unfolded proteins. An increase in unfolded proteins or a decrease in proteasome activity can result in protein aggregates. So this figure down here is actually what you will have in your slides, like maybe a couple of minutes from now. I'll go over it with you again then, but just for a very quick overview, at the bottom we have increasing time. With a newly synthesized protein, what happens very often is that the quickest thing that happens is that it gets correctly folded without any help. Sometimes the protein is still unfolded, upon which you will hopefully get it correctly folded with the help of a molecular chaperone. Sometimes it is still unfolded. In that case, you have to get rid of the unfolded protein. The proteasome will help you do that. If you still do not get rid of the unfolded protein, what can happen is the protein can form aggregates. Imagine the protein's unfolded, it has all these hydrophobic residues exposed. It's going to form aggregates, clumps with other proteins. It's like a pile of garbage in your cell. It can be a pile of garbage around your cell. It's really going to hurt function. And so one thing I really want to show you is that students will say, okay, we understand the proteasome. At the end of the class, we'll understand the proteasome. We'll understand unfolded proteins. But when you have these piles of garbage, how come you get these types of symptoms? So just to show you how you get those symptoms from these piles of garbage, from these unfolded proteins, let me show you a couple of minutes from this video. It's about Alzheimer's disease. Think of the plaques and tangles as these piles of garbage in your cell and around your cell.
What is Alzheimer's disease? Alzheimer's is a slow, fatal disease of the brain affecting 1 in 10 people over the age of 65. No one is immune. The disease comes on gradually as 2 abnormal protein fragments called plaques and tangles accumulate in the brain and kill brain cells. They start here in the hippocampus, the part of the brain where memories are first formed. Over many years' time, the plaques and tangles slowly destroy the hippocampus, and it becomes harder and harder to form new memories. Simple recollections from a few hours or days ago that the rest of us might take for granted are just not there. After that, more plaques and tangles spread into different regions of the brain, killing cells and compromising function wherever they go. This spreading around is what causes the different stages of Alzheimer's. From the hippocampus, the disease spreads here to the region of the brain where language is processed. When that happens, it gets tougher and tougher to find the right word. Next, the disease creeps toward the front of the brain, where logical thought takes place. Very gradually, a person begins to lose the ability to solve problems, grasp concepts, and make plans. Next, the plaques and tangles invade the part of the brain where emotions are regulated. When this happens, the patient gradually loses control over moods and feelings. After that, the disease moves to where the brain makes sense of things it sees, hears, and smells. In this stage, Alzheimer's wreaks havoc on a person's senses and can spark hallucinations. Eventually, the plaques and tangles erase a person's oldest and most precious memories, which are stored here in the back of the brain. Near the end, the disease compromises a person's balance and coordination. And in the very last stage, it destroys the part of the brain that regulates breathing and the heart.
Okay, so I'll stop it there, but hopefully you can get an idea of how this really simple concept, right? Unfolded proteins, the proteasome not working, can kind of start to accumulate the variety of symptoms you see in a whole bunch of different diseases. So what I want to kind of emphasize here is that this lecture really lays the foundation for a lot of upper year topics and a lot of things we talk about when we discuss a whole variety of diseases, including how we'll discuss Alzheimer's and cancer a bit more in this particular course. Now, don't forget, maybe in high school, first year bio 130, and even a lot of bio 230, we talk a lot about transcription, a lot about translation. We talk about how you make proteins, but don't forget, getting rid of the proteins can be just as important. And we're going to talk about that in this particular class. So here is the outline slide that we've been showing you every time you've been here. Again, you cannot change your DNA sequences very well. You can change what comes out of your DNA. You can change DNA organization, change transcription. Post-transcriptional modification of RNA is possible, splicing modification is possible. In this particular class, we'll talk about translational modification, and I'll talk about post-translational modification as well. Now, this is a review slide from the very first lecture you were here. In the very first lecture, we said that a 2D gel gives you a snapshot of the proteome. So a 2D gel, we said, gives you a snapshot of the proteome. So I see a lot of people writing when you wrote it down in the first class, right? 2D gel. Gives you a snapshot of the proteome. And in the very first class, we showed you this, and we said what we're looking at here are two 2D gels. So we can kind of divide it in half here. We have the human brain on the left, 2D gel for the human liver on the right. And remember last class we said that DNA microarrays, you kind of read them like a table. For 2D gels, remember, you kind of read them like a graph. On the y-axis is molecular weight going from low to high. And on the x-axis is the isoelectric flux going from acidic to basic. Same thing for the human liver, right? Again, on the y-axis is molecular weight. On the x-axis is the isoelectric point going from acidic to basic. And we said that, well, normally your 2D gels are not going to be color coordinated for you, but in your textbook they've color coordinated these ones. We said that red are the blobs that are common to both, the proteins that are common to both, and blue are the blobs or the proteins that are tissue specific. So we can look at any two of these. Let's say we'll look at this one right here. You can see it's fairly high in molecular weight, also fairly acidic in isoelectric points. And the same one here is in the liver. Again, fairly high in molecular weight, fairly acidic in isoelectric points. And in the very first class we said, yes, the human brain and the human liver, they can have the same protein. But look, in this case, they're here at different amounts. The red blob is much smaller here. It's much larger here. You can have the same proteins. They'll be there in different amounts. As well, you have some proteins that are unique. So in blue here, you can see this one again is a little bit lower in molecular weight, a little bit more acidic, and that blue blob is missing in the human liver. So you have some proteins that are unique, that are going to be in brain, not in liver, and vice versa. Again, 2D gel, read it kind of like a graph. Some proteins will be common to both the brain and the liver, but you'll have them in different amounts, possibly. As well, you have some proteins that are unique. Both prokaryotes and eukaryotes use translational control mechanisms to regulate protein expression, often in response to stressful situations such as low nutrients, infection, or environmental stresses. This is similar for both eukaryotes and prokaryotes, but we're going to start off with a prokaryotic example and some prokaryotic mechanisms. In prokaryotes, their mRNAs have a 6-nucleotide Shine-Dalgarno sequence. You want to be a little careful here. That's in prokaryotes, not eukaryotes. So as it says here, prokaryotes, not eukaryotes, have a 6-nucleotide Shine-Dalgarno sequence. They're essentially just named after the scientists who discovered them. So there's a Dr. Shine and there's a Dr. Dalgarno. It's a sequence upstream of the AUG start codon. It's before the start codon, typically about 8 base pairs or so before. Around 8 base pairs or so before. And the prokaryotic mRNAs need this Shine-Dalgarno sequence. It's before the start codon. They need it because it's going to help correctly position the start codon in the ribosome and provide translational control mechanisms. If you do not have access to the Shine-Dalgarno sequence, the prokaryotes will not make the protein. You may make the protein in very little amounts, but you probably won't make much of it at all. So you need access to the Shine-Dalgarno sequence for translation. A couple of examples we'll go through. The first one is RNA binding proteins. They can actually block access to the Shine-Dalgarno sequence. In blue, you have your standard setup for your prokaryotic mRNA. You should be able to tell it's prokaryotic mRNA because, well, there's no poly A tail, there's no 5' cap. And in orange here, there's an SD sequence. That's our short form for Shine-Dalgarno sequence, right? Only in prokaryotes. Now you can map out your mRNA. So again, remember, from the start codon, from the AUG, going to the stop codon, that is your coding sequence. So CDS, I'll write in the coding sequence. Starting at the start codon, ending at the stop codon. Now you'll have other regions that are transcribed but not translated. So they're not coding for anything, but they're still transcribed and they're quite important. Regions up here include the 5' UTR, you'll remember, so 5' untranslated region. And then after the start codon will be a 3' untranslated region. So again, those are transcribed but not translated. Okay, so again, showing you your general setup. This is a prokaryotic mRNA, no cap, no poly A tail. Coding sequence starts at the start codon, ends at the stop codon. 5' UTR, 3' UTR, untranslated region. CDS, that's the coding sequence. Now, it's within the 5' UTR that you have a Shine-Dalgarno sequence. It doesn't code for anything, but it's quite important. So you can see in this case, the Shine-Dalgarno sequence is exposed. There's no issue here. The ribosome can be correctly positioned. The mRNA can be correctly positioned in the ribosome. You will have translation, no issue. Now, in the presence of a translational repressor protein, what can happen is a translational repressor protein can bind to the 5' UTR. It blocks access to the Shine-Dalgarno sequence. The ribosome cannot get access to the Shine-Dalgarno sequence. It will not be correctly positioned. You will not have translation. So notice again that those uncoding regions are still important. They don't code for protein, but they're very important. Okay, so again, in the 5' UTR, you have a Shine-Dalgarno sequence. Right now it is exposed, no problem. The ribosome can bind to it. It can be correctly positioned. You get translation. If you have a translational repressor protein binding to it, well, now you'll block translation. Now, note here, this is a translational repressor protein. Last class we talked about splicing repressors. Class before that we talked about transcriptional repressors. Do not confuse them. You want to make sure you compare them. Compare this to splicing repressors to transcription repressors. Don't mix them up. A second mechanism by which you can control these Shine-Dalgarno sequences or use them for control is in temperature-regulated RNA structures. So this example is from Virulence genes of the human pathogen Listeria monocytogenes. So I'll tell you a bit about Listeria. You don't have to write this down, it's not on the exam, but it's kind of interesting. Just of course, if you get infected by Listeria, of course you get really sick. It affects primarily people with weakened immune systems, so think newborns, pregnant women, and the elderly. About a quarter of the people who get infected with Syria actually die from it, and they die from meningitis and sepsis. So meningitis is inflammation of the membranes surrounding the brain and the spinal cord, and sepsis, you can think whole body inflammation. Now, what's quite interesting is that Listeria actually uses the Shine-Dalgarno sequence for regulation, but it uses this whole region in a thermal switch. It's like a thermostat. Like with the right temperature, translation turns off. Right temperature, translation turns on, right? You get all those different elements. So your setup starts out the same way. Again, from the start codon to the stop codon, that's the coding sequence. And again, before the coding sequence, say this region here is the 5' UTR. And then the region after the start codon, or after the stop codon, is the 3' UTR. And again, your setup's the same. Coding sequence from the start codon to the stop codon. 5' untranslated region before it. The 5' UTR includes the Shine-Dalgarno sequence. 3' UTR after. Now, at lower temperatures, what's happening here is the 5' UTR, including the Shine-Dalgarno sequence, forms this stem loop structure. So here is the stem portion, right? And here is the loop. And you can see what's happening in the stem portion is that you have a lot of hydrogen bonding. The RNA is base pairing with itself. It's anti-parallel and complementary. And what's happening here is the Shine-Dalgarno sequence is blocked. The ribosome cannot access it. You cannot correctly position the mRNA. Translation is on. What can happen at increased temperature, in this case it's going to be 37 degrees, it'll be different for different RNAs, but in this case with 37 degrees Celsius, you can break those hydrogen bonds, you will now be able to access the Shine-Dalgarno sequence, this prokaryotic mRNA will be translated. So again, it's kind of like a thermostat, it's like a thermal switch. At the right temperature, you break these bonds, Shine-Dalgarno sequence can be accessed, you can get this thing correctly positioned, you'll get translation. Cooler temperatures you will not get. Now, I know I get a lot of questions about the thermal switch and these types of RNA structures, and we get a lot of these questions in the morning section as well. So what we did was we added this abstract in your notes. So we're not going to test you on this particular abstract, but it is quite interesting. So I would encourage you to take a look at it if you're so interested. We'll also post the paper online. This is about a particular bacteria that causes meningitis. So again, think of inflammation of the membranes surrounding the brain and spinal cord. And this particular bacteria actually uses 3 separate RNA structures, 3 separate thermal switches, just like we talked about on the last slide here. And these 3 separate RNA switches using temperature helps trigger immune evasion. So it's actually pretty cool stuff. I would encourage you to take a look at that as we go. Okay, the third mechanism by which you can control translation via the Shine-Dalgarno sequences is via a simple riboswitch. I won't draw it in for you again because it's actually quite simple. It's the same setup from the start codon to the stop codon. That's the coding sequence. Before that is the 5' untranslated region. Within the 5' untranslated region, you have the Shine-Dalgarno sequence. You can see in this case, the 5' UTR may have a structure, but in this case, you're not blocking the Shine-Dalgarno sequence. This RNA can be correctly positioned. You will get translation being on. Our ribosomes can bind to it, translation gets done. With a small molecule, you can get structural rearrangement of the RNA blocking the Shine-Dalgarno sequence. So here's a small molecule, could be S-adenosylmethionine, could be another molecule, depends on the RNA. In this case, you can get rearrangement of it. Now you will block access to the Shine-Dalgarno sequence. Translation will be on. Similar to the riboswitches we talked about in previous lectures. Now be careful though, with the purine bioswitch, with the purine riboswitch, that terminates transcription. Here we're talking about translation. So transcription, translation, right? They're different. Purine riboswitch, that was transcription. This is translation. Last mechanism we'll talk about is antisense RNA. Your setup again starts out being the same. In the 5' UTR, you have a Shine-Dalgarno sequence. If nothing is blocking it, our ribosome can bind, you can get translation, no problem. What can happen is the prokaryotes can produce antisense RNA elsewhere from the genome, and this antisense RNA produced elsewhere in the genome is going to base pair. Look at base pairs anti-parallel and complementary. That will turn translation off. You're blocking access to the Shine-Dalgarno sequence. Notice in this case, this would be a double-stranded piece of RNA. Right? Now again, you want to be a little careful here, because here we're talking about prokaryotes. We're talking about antisense RNA. Last class we talked about microRNAs and eukaryotes. Do compare and contrast. Last class was eukaryotes and microRNA. This is prokaryotes and antisense. So in a later tutorial, not this one, but a later tutorial, we'll show you some questions comparing those two. So you do want to go back and compare them. Now eukaryotes, again, don't have Shine-Dalgarno sequences. Eukaryotes do not have Shine-Dalgarno sequences, but they can have similar mechanisms as eukaryotes. You can have translational repressors that bind near the initiator AUG and inhibit translation, say in aconitase. Now to understand the figure on the left here, what you want to do is first look at the first 2 points. Ferritin binds iron and releases it in a controlled manner. It is not needed when iron is low. Okay? So again, ferritin binds iron and releases it in a controlled manner. It is not needed when iron is low. In the first case, you have iron starvation. So iron is low, right? So that means you do not need ferritin. Here is your general setup again. Now this is in eukaryotes, so you should have a 5' cap. It's not drawn here for you, but you should have a 5' cap. You have a poly A tail. In yellow on this side is the 5' UTR, 5' untranslated region. In red is the coding sequence. And then over here in yellow is the 3' untranslated region. You can see when you have iron starvation, again, you don't have ferritin, right? So this is the ferritin mRNA. What's happening here is cytosolic aconitase is binding to the 5' UTR. If you remember your translation initiation from last class, remember what has to happen is the small ribosomal subunit has to start at the 5' end and look for the start codon. If cytosolic aconitase is here, the ribosome cannot get past it. You cannot find the start codon. So there's going to be no ferritin protein. All right, so let me repeat that. Again, with cytosolic aconitase binding to the 5' UTR, well, the small ribosomal subunit, it cannot get past that region. It cannot get past it to find the start codon. So that means you'll get no translation, no ferritin will be made. Now you have the other option of excess iron. So if you have a lot of iron around, well, iron will bind to cytosolic aconitase. Cytosolic aconitase will release the 5' UTR. And if you release the 5' UTR, well, now the small ribosomal subunit can get through this, can find the first AUG, and you'll get ferritin protein being made. So as it says, when ferritin is low, aconitase binds to the ferritin RNA near the start site and blocks translation. With excess iron, you will get translation. Aconitase binds to the iron, exhibits a conformational change, and the ferritin RNA is released. Right, so again, very quick summary. With low iron, iron starvation, aconitase binds, small ribosomal subunit cannot find the AUG, no ferritin protein is made. With excess iron, you will release it. Now the small ribosomal subunit can get through and find the AUG. Now recall, we talked about aconitase last class with the transferrin receptor. So that's something else that you want to compare and contrast. Cytosolic aconitase binds to the 3' UTR of the transferrin receptor mRNA. But here it binds to the 5' UTR of the ferritin. So do make sure you compare and contrast. Remember last class I told you to memorize that and come in with that. So again, last class, transferrin receptor, 3' UTR. Now, eukaryotes also have other mechanisms. You can interfere with the 5' cap and poly A tail binding, as well you have those microRNAs. So last class, we said that your mRNA here is not broken, right? It's just really long. That's why they draw it with these dashed white lines. It has a cap, has a poly A tail. We said that to begin translation, you need to form this loop. EIF4E binds to the cap, EIF4G binds to that, poly A binding proteins bind to that, poly A binding proteins also bind to the poly A tail. You need this loop to start translation. Well, if you want to stop translation, you can interfere with this loop, stop the loop from being made. As well, you have other mechanisms like microRNAs in eukaryotes. So again, remember, compare the microRNAs to the antisense we talked about in this class. There's also other eukaryotic specific mechanisms. What we want to focus in on this particular class is EIF2. So EIF, that's eukaryotic initiation factor. See that on the exam, you know right away, eukaryotes, it's the E. This is your setup from last class. Just a quick review, because you've seen this last class. This is the small ribosomal subunit. Before you have the large one around, before you have the mRNA around, you already have the small ribosomal subunit. You have the initiator tRNA. This is the one that binds to the AUG, has methionine on it. You have EIF2, what we're going to talk about now, bound to GTP. You need this to start the whole process. Separately, you have your mRNA in a loop. The small ribosomal subunit will start at the 5' end and will search for the first AUG. What you want to focus in on, again, is EIF2. It plays a crucial role in translation initiation. You need it to form that complex with GTP and the initiator tRNA. So you need EIF2 bound to GTP to start this whole process. Now, the small ribosomal subunit is going to search for the first AUG. When it finds it, GTP will be cut to GDP, and then EIF2 is going to be released. So again, you're here, right? We're here. We're searching for the first AUG. You're using ATP to find the first AUG. When you find the first AUG, the initiator tRNA will bind to it. EIF2 will cut the GTP into GDP. Then EIF2 bound to GDP will be released. That's when the large ribosomal subunit can come in and start. Okay, so at the end of this, EIF2 cuts GTP to GDP, gets released, large ribosomal subunit binds. Now, think about what happens with EIF2. At the beginning of the process, so at the beginning of the process right here, you need EIF2 bound to GTP to start the whole process. At the end of the whole process, you have EIF2 cutting GTP to GDP, and then that goes away. So if you want to restart translation, if you want to initiate another round of translation, you have to get the EIF2 that's bound to GDP to bind to GTP, right? EIF2 bound to GDP is not useful. You need it to bind to GTP. How do you do that? How do you get rid of the GDP and bind to GTP? Well, it's EIF2B that does it, and it mediates a guanine nucleotide exchange. So you want to be a little careful here. If you look at what's happening here, you have EIF2B, which is a guanine nucleotide exchange factor. It's an exchange. It's not a phosphorylation, right? So you're exchanging GDP for GTP. Yes, this is guanosine diphosphate. Yes, this is guanosine triphosphate. But it's not like you're phosphorylating the GDP for GT. It's not a phosphorylation, it's an exchange. Let me show you this in the figure. Here is inactive EIF2, right? EIF2, it is bound to GDP. That's not useful for you starting translation. So you're going to use EIF2B. EIF2B will bind to the EIF2 and mediates an exchange, releases the GDP. You'll then bind to the GTP. Now you have active EIF2 bound to GT. Now you can start translation again. Okay, let me review very, very quickly because we add another layer of complexity onto this. At the beginning, EIF2 bound to GTP. After you start translation, right, after you bind to the AUG, EIF2 bound to GTP, we'll cut it to GDP. Then you'll start translation. Now EIF2 is bound to GDP, but you can't start another round of translation with this. So what you're going to do is you're going to mediate an exchange, not a phosphorylation, but an exchange. So EIF2B will cause the release of GDP. You'll get GTP binding. Now you can start another round of translation. So this right here is an exchange, but this is further complicated because EIF2 itself can be separately phosphorylated. Separately phosphorylated. So this is what we show you by the phosphorylation. This is what we mean by the phosphorylation. Now, if you look here, this is EIF2 bound to GDP. Say the cell wants to shut down translation. Maybe there's not enough nutrients around, maybe it's made too much protein, whatever. It wants to shut down translation. So if the cell wants to shut down translation, it can activate a specific protein kinase that phosphorylates EIF2. So here's EIF2, it's still bound to GDP. Separately, it is phosphorylated. What's going to happen now is EIF2B will try to mediate the exchange, but it can't. If you have the phosphorylation, there will be no exchange. So you will not start translation again. On top of that, there's in fact a lot more EIF2 than EIF2B in cells. There's actually very little EIF2B in your cell. So if your cell really wants to shut down translation, what it can do is just phosphorylate a couple of EIF2s. And because you have so little EIF2B in your cells, if you just phosphorylate a couple of EIF2s, they'll bind to the EIF2Bs, and all your EIF2Bs will be stopped. They won't be able to mediate exchange of the other EIF2s. There's very little of that stuff around. The EIF2 that's phosphorylated is almost like a sponge, soaking that stuff up. And so again, there's very little EIF2B in your cells. If you want to shut down translation, if you just phosphorylate a couple of these, the very few EIF2Bs that you do have will be stopped in this state. They won't be able to mediate exchange of anything else. They're just stopped. Translation will shut down. Now, say you shut down translation, a lot of you will ask, and get this question kind of all the time at the breaks, well, if you shut down translation, won't the cell die, right? The cell needs proteins to live. Well, not all mRNAs are equally affected by EIF2 phosphorylation. The couple of mRNAs that are essential, they will not be shut down in this manner. And a couple of mRNAs that are essential, they'll have other mechanisms to continue on with translation. All right, so let's go back to the outline. We talked a little bit about translational mechanisms. We talked about how Shine-Dalgarno sequences are used in prokaryotes. Eukaryotes have other mechanisms. You have aconitase, microRNAs. You can interfere with a loop. You have EIF2B. Now we're going to talk about post-translational modification of proteins. So this gets into what I kind of talked about in the preamble with protein unfolding, the proteasome diseases like Alzheimer's disease. We'll see a little bit about cancer at the break. For proper protein folding, you'll often get covalent modification involved. You'll often get protein interactions. with other subunits. Now the text really just describes what's on the figure here. Here's a new polypeptide chain. Typically, you'll have folding and cofactor binding occurring quite rapidly, so the cofactors could be stuff like iron, zinc, etc. You'll often have covalent modification by glycosylation, phosphorylation, acetylation, etc. Here's a phosphate group that's added. This could be an acetyl group. And sometimes you'll binding to other protein subunits, and that will give you a mature functional protein. For protein folding, hydrophobic amino acids are typically buried in the interior core. They're not surface exposed. And protein folding usually begins somewhat, at least, as the polypeptide is emerging from the ribosome. First point's not too difficult to understand. If you have hydrophobic amino acids, they're typically going to want to get away from the water. You have lots of aqueous solution in the cell, so that's going to want to get away. You have to get into the interior core of the protein. And typically, as you're making the protein, so the MR is in blue here, this is the ribosome, as you're making the protein, it'll start to fold. You may have different domains on a protein. Some polypeptide chains have one domain, some have multiple domains. You can see folding of different domains here. Now this is kind of the initial view that we always talk about, but in fact, most proteins do require help to fold. They require other proteins called chaperones. There are these proteins called chaperones that help other proteins fold. Now, many chaperones are called heat shock proteins, HSPs for short, but you want to be careful here. It says here they are synthesized to high amounts by cells at elevated temperatures. So, yes, if you heat the cell, you'll have more heat shock proteins. And that is somewhat logical, right? If you heat the cell, you're breaking all sorts of bonds. You'll typically break non-covalent bonds. Well, you're going to need more heat shock proteins to help those proteins fold back up. If you're destroying their structure upon heat, you're going to get more chaperones around to help them fold back up. So yes, you'll have more of these heat shock proteins at elevated temperatures, but they're still there at regular temperatures. Still there at regular temperatures. These are just typically named after how you discover them. So many heat shock proteins are so named because scientists were looking at proteins, genes overexpressed when you heat the cells, and they found these proteins. So that's kind of how they're named. But they're definitely there at regular temperatures as well. Two examples are HSP70 and HSP60, so heat shock protein, and then the numbers, typically the molecular weight, 70 versus 60. HSP60 is chaperonin. It looks a little bit confusing here. Some students think that we're saying both of them are chaperonin, and no, HSP60 is really the chaperonin. They do function somewhat similarly in that they both interact with exposed hydrophobic residues of misfolded proteins. Both use the energy from ATP hydrolysis to promote proper folding. So if I just kind of zoom up on the figure here, you can see how HSP70 works. Now, how HSP70 works is really quite complicated. You do have to understand all sorts of thermodynamics, and there's a lot of biophysical calculations to this, but I'll give you kind of the general overall view. So, say the ribosome is making this polypeptide chain in dark green. As these hydrophobic amino acids are exposed, the HSP70 machinery will bind to it. And then using ATP, it's going to change the polypeptide a bit. It might release it upon the right time. It's all about timing. At the right time, it's going to release the right amino acid so that they will adopt a proper structure. So that's kind of the summary of how it works. Now, hopefully you will get a correctly folded protein. Sometimes you'll still have an incorrectly folded protein. That's when you have this HSP60-like protein coming in. This one is much more easy to understand. It essentially looks like two barrels, like a double barrel structure. You have this hydrophobic protein binding site at the end, so it's going to find incorrectly folded proteins if they have their hydrophobic amino acids exposed. You'll bind to it, it'll feed it into the chamber. It's going to use ATP, it might change shape a bit, grow a bit, get a bit larger, it'll have a separate cap, and essentially it functions like an isolation chamber. Gives this polypeptide time to fold. You don't want it to start forming aggregates. You don't want it to start binding to hydrophobic amino acids of other polypeptides and clumping up. Just form an isolation chamber, give it more time to fold. Then with some more ATP, you'll get the release of a cap, and you'll get the release of a correctly folded protein. So again, the top one, HSV70, really there's timing to it, binds to hydrophobic amino acids and releases it at the right time, uses ATP. Bottom one, like an isolation chamber, also uses ATP, but really just gives it more time to fold. Now, improperly folded proteins obviously can aggregate and form those piles of garbage we talked about. So improperly folded proteins, they can aggregate, they can become toxic to cells. Misfolded proteins are the cause of many inherited human diseases, and the process is closely monitored by a protein-degrading apparatus called the proteasome. Exposed hydrophobic residues are marked for protein degradation by the proteasome, and it'll compete with chaperones for misfolded proteins. Chaperones try to fold it back up, proteasomes destroy it. This is the slide I showed you from the preamble. This is increasing time. A newly synthesized protein, the quickest thing that can happen is you can get it correctly folded without any help. Imagine as soon as it emerges from the ribosome, it's folded. Sometimes it'll still be unfolded, in which case you need a molecular chaperone, like those heat shock proteins, to help it fold. Sometimes it's still unfolded. If it's still unfolded, well, you better get rid of it with the proteasome, destroy that protein. If it's still unfolded, well, now you have a pile of garbage, right? You have protein aggregates. That is not good. The longer time it takes to fold, the more chance you have of being degraded. To destroy these proteins, you use the proteasome. It's in both the cytosol and the nucleus. So this really emphasizes how important the proteasome is. 1% of all your cellular protein is the proteasome. 1%, you make a lot of proteins. 1% of your protein is devoted to destroying proteins. So this is a very key aspect of many cellular processes. It's a key aspect of the cell. It's a hollow cylinder with a cap at each end and an active site in its core. It's going to protect cellular proteins from degradation. You're not going to degrade everything. You're going to degrade specific proteins. This is what it looks like. Here is the cylinder. In red here, this is the active site. You can see there's a cap on each end. The cap is only going to let specific proteins through. You're not degrading everything. You're degrading specific stuff. The proteasome is going to act on proteins that have been marked for degradation by a small protein tag called ubiquitin. You're only degrading proteins with a ubiquitin. Be a little careful here, you'll see this gets a little bit confusing. Ubiquitin itself is a protein. Ubiquitin is a protein, and it marks other proteins for destruction. If you take a look at the cartoon diagram, you can see this is your standard proteasome. You have a cap at each end, because again, you're not degrading everything, you're degrading specific stuff. Here is the central cylinder, here's the active site. It's like a pencil sharp, just destroying polypeptides that come in. Here is the green protein that you are degrading. Now, you're not degrading just any protein, you're degrading proteins that have been marked with the ubiquitin. Take a look at how you're marking this protein. So this green protein has this red ubiquitin on it. So this red light bulb thing, that's a ubiquitin. Now, not only do you have one ubiquitin on it, you have one red light bulb ubiquitin. On that, you have another ubiquitin. On that, you have another ubiquitin. On that, you have another one. It's what we call a polyubiquitin chain. Not just one, polyubiquitin chain. The cap will recognize that polyubiquitin chain. You can recycle those ubiquitin proteins. You won't destroy those ubiquitin proteins. They're going to be fine. You will destroy the green protein that was marked, right? Chop it all up. That's it. So, quick review. It gets a little bit more complicated. Here is the proteasome. You have two caps. You're only going to let polyubiquitinated proteins in. Central core to destroy the proteins. This green protein has been marked for destruction. It's been marked for destruction by having a red protein called a ubiquitin on it, and on that red protein called a ubiquitin, you have another ubiquitin. On that, you have another one. On that, you have another one. It's a polyubiquitin chain. The cap will recognize that. You'll reuse the ubiquitin, and you'll destroy the polypeptide chain. Now, this is the general overview of a process, right? You have a particular protein, you mark it for destruction with that polyubiquitin chain. The next couple of slides are talking about how you add this polyubiquitin chain. This is how you add the polyubiquitin chain. Before we kind of start this, I just want to mention one thing here. So, I did not learn, to be honest with you, I did not learn about this until maybe second year graduate school, and you're learning about this in second year undergraduate. And I really did not kind of understand it until maybe fourth year graduate school, and I want you all to understand it now in second year undergraduate, right? So we're going to go slow, make sure you take lots of notes, go back and read the textbook, and then ask me questions at the break in the third hour. Okay? So it's a little bit complicated, but we'll take it one step at a time. So to add, right, we're adding that polyubiquitin chain. To add that polyubiquitin chain, 3 enzymes are involved, an E1, an E2, and an E3. The names of them are here. So this is the E1, this is the E2, and this is the E3. So E1, you just wrote down the names, right? E1's an ATP-dependent ubiquitin activating enzyme, creates an activated E1-bound ubiquitin. E2 is a ubiquitin conjugating enzyme, and then E2 and E3 is what we call ubiquitin ligase. So this will become a little bit clear. One thing to note, E2 and E3 form a complex. That's what we call a ubiquitin ligase. It's both of them together. The E2-E3 complex is called a ubiquitin ligase. Make sure you go back to read the textbook. The textbook talks about this a little bit and says how if you read scientific literature, often they're quite flexible. There's a little bit of flexibility in the terminology. And sometimes people will just call the E3 a ubiquitin ligase, but more traditionally and more properly, it's an E2-E3 complex. It's those together that we call a ubiquitin ligase. Okay, so let me take you through this figure step by step. So the text here becomes quite clear if I show you on the figure. E1 here is a ubiquitin activating enzyme. So it's going to take this ubiquitin protein using ATP. It's going to add the ubiquitin to itself. It's going to be activated. It's going to load itself with the ubiquitin. Now separately, you have the ubiquitin ligase, right? So that's E2 bound to E3, ubiquitin ligase. E1 that's been loaded with the ubiquitin will now bind to the ubiquitin ligase, and then will transfer the ubiquitin over to the E2. And this ubiquitin ligase, as it says here, is primed with ubiquitin. So it's now the E3 here, this section, that will recognize a protein, and then the E2 will put the ubiquitin on the protein. Okay, so again, E1 right here. There are a lot of people still writing this. Okay, so again, E1 is ubiquitin activating enzyme. Takes that ubiquitin protein, using ATP, adds it to itself. Then E1 will bind to the ubiquitin ligase, that's E2 bound to E3. and the E1 will transfer the ubiquitin onto the E2. Now the E3 is going to bind to a specific protein, and the E2 will add the ubiquitin onto that protein. Now, one thing to know, which I found really confusing in graduate school, I didn't really quite get this, was that, and this is a good thing to write down, in humans there are a couple of different E1s. In humans there are a couple of different E1s. There's roughly 30 E2s. There's roughly 30 E2s. And there's hundreds of E3s. They'll all interact with specificity. They'll all interact with specificity. So again, few different E1s in humans, 30 different E2s or so, hundreds of different E3s. They'll all interact with specificity. So some E1s will interact with some E2s, which will interact with some E3s. All sorts of different combinations. And then different ubiquitin ligases will recognize different proteins. They'll all kind of behave differently. Now to add it, the E3 is going to find specific degradation sequences. And the whole process repeats itself. So here is a protein that you're going to degrade, and this is a particular lysine on that particular protein. Now again, you're not degrading just any protein, you're degrading proteins that are kind of ready for degradation. So this one will have a particular degradation signal. We'll talk about this in a sec, but maybe you have hydrophobic amino acids exposed, some type of signal that says, oh, this protein should be degraded. So you can see the E3 portion will bind to that particular degradation signal, and then ubiquitin will transfer over from the E2 over to the protein. Whole process repeats itself many, many times, right? The E1 comes in again, and you'll have this polyubiquitin chain. Now, with this polyubiquitin chain, you'll be degraded by the proteasome. So very, very quick overview. Again, we started off with this proteasome. You're not destroying every protein. You're destroying specific proteins with this polyubiquitin chain. To add that polyubiquitin chain, you have an E1 adding into itself, then binding to the ubiquitin ligase, and then ubiquitin gets transferred to the E2. The E3 will bind to a specific protein sequence, protein degradation signal, and then the ubiquitin will transfer over from the E2 over onto the protein you're degrading. Whole process repeats itself many times. You get a polyubiquitin chain, and then you get degradation. Okay? So that's kind of the general process so far. Now, you don't have 2 slides. I just want to show you this. This is ubiquitin. It is a protein. It is a protein. And you're not adding ubiquitins just anywhere. You're adding them onto specific lysine residues. This is a lysine amino acid. This would be the carboxyl carbon. This is the amino. This is the alpha carbon, right? Here's the R group. You can see carbon, 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 carbon. And then it's on this NH2 that you stick on the ubiquitin. And just to really emphasize the importance of this thing, again, in first year, second year, we really concentrate on transcription and translation and making proteins. But getting rid of your proteins can be just as important for a number of different diseases. And actually, for this type of a mechanism, for ubiquitin-mediated protein destruction, these 3 scientists were actually awarded the Nobel Prize in Chemistry in 2004. So I know it's Nobel Prize week, right? Lots of Nobel Prizes being announced, and these guys actually won for ubiquitin a couple of years ago. Now, the ubiquitin modifications can actually have all sorts of other functions, not just in degradation. And sometimes it depends on how they're added. Sometimes it depends on how many are added. So if you take a look here, this is what we call monoubiquitination. Imagine you have a protein, if you just add one ubiquitin on it, typically those types of proteins are involved in histone regulation. This is what we call multi-ubiquitination. This is multi, not poly, right? That's different. So this is multi-ubiquitination. Here's one protein, you get 3 separate ubiquitins, right? Different ubiquitins on different locations, or I should say ubiquitin on different locations. This protein will typically be involved in endocytosis, and this is something we're going to talk about in the second part of the course. Okay? So make a note later on to talk about this. Polyubiquitination is just what we went over. Here is a protein, and you have one ubiquitin on another one, on another one, on another one, on another one. You have to be careful of the linkages here, because depending on how they're linked, the protein could be degraded or could be involved in DNA repair. Now it says here, the linkage occurs through lysine 48 of ubiquitin. Now on this green protein here that you're degrading, you'll have many amino acids. You'll have many lysines. On a particular lysine, doesn't have to be lysine 48, on a particular lysine, you add that ubiquitin. And now this ubiquitin, on its lysine 48, you'll add another ubiquitin. And then on this ubiquitin's lysine 48, you add another ubiquitin. So on and so forth. Let me repeat that. This green protein, many different amino acids, many different lysines. On a particular lysine, does not have to be lysine 48, on a particular lysine, you add a ubiquitin. Now, on this ubiquitin's lysine 48, you add another ubiquitin. And then on this ubiquitin's lysine 48, you add another ubiquitin. So on and so forth. And that will signal for proteasome degradation. If the linkage is through lysine 63 of ubiquitin, it might be involved in DNA repair. So the general setup is, again, quite similar. This protein will have many different amino acids, of many different lysines. On a particular lysine, does not have to be lysine 63, but on a particular lysine, you have a ubiquitin added. And then on this ubiquitin's lysine 63, you have another ubiquitin. And on this ubiquitin's lysine 63, you have another. Okay? So the linkages here are talking about the linkages on the actual ubiquitin. Depending on the ubiquitin ligase, you can modify it, you can control it through all sorts of different mechanisms. Again, there are all sorts of different ubiquitin ligases. We said there's maybe 30 or so different E2s, hundreds of different E3s. You get all sorts of different interactions. Different ubiquitin ligases will be controlled differently. So for some, maybe the ubiquitin ligase is inactive. Then upon phosphorylation by a specific protein kinase, now it's active. Sometimes it'll be inactive and you'll need allosteric transition caused by a ligand binding. This particular ligand, nutrient, waste, whatever, comes in and binds, now it will be active. For some, they'll be inactive, and you'll need another subunit to get it to activate. It really depends on things. Now that's for the ubiquitin ligase. For the proteins that you degrade, it can be activated upon different ways as well. So you can see sometimes with proteins you're not going to degrade them. Then sometimes with phosphorylation by a protein kinase, now it's a signal to be degraded. Now it will be bound by a ubiquitin ligase. Sometimes you'll need to remove a subunit, and then that will expose a signal to be bound by ubiquitin ligase. Sometimes the protein will be cleaved, and then you'll get binding by ubiquitin ligase. Really depends on that. Okay, so with that being said, what I want to do is I want to leave this quicker question on for the break. We're going to take a 10-minute break. Again, questions are welcome, and we'll take this up after the break, okay?